Hydrogen from biomass feedstock. Feedstock used. A wide range of biomasses including agricultural crops, their residues, lignocellulosic biomasses, aquatic biomasses such as algae and water weeds, industrial or municipal solid waste and animal waste that can be utilized for the production of biohydrogen. These feedstock can be divided into the following categories. Energy crops includes herbaceous crops, woody crops, industrial crops, agriculture crops and aquatic crops. Agriculture residues and waste. Crops obtained during crop harvesting and processing and animal waste obtained during cattle farming. Forestry waste and residues includes wood residues from mill, log residues, trees and shrub residues. Industrial and municipal waste includes industrial solid waste, sewage sludge and industrial waste. The feedstock can be classified into first, second and third generation crops. First generation biomasses includes agriculture crops. Second generation biomasses include lignocellulosic biomasses and third generation biomasses include algae. Second and third generation biomasses are commonly preferred for biohydrogen generation since they are non-food crops. Methods involved in biohydrogen generation. The following are the major methods involved in biohydrogen generation using biomass as a feedstock. First, thermochemical methods. The thermochemical method of biohydrogen production mainly includes pyrolysis and gasification. Pyrolysis. The pyrolysis involves the heating of biomass at a temperature ranging from 650 to 800 degree Kelvin in the absence of oxygen. Bio-oil, charcoal and gaseous compounds are the major products of biomass pyrolysis. Fast pyrolysis is usually considered for hydrogen production since it generates more gaseous products than slow pyrolysis. Slow pyrolysis yields more of charcoal, that is rich carbon content. The following equation represents the reaction involved in the fast pyrolysis. Biomass heated produce hydrogen, carbon monoxide, methane plus other products. Methane and other hydrocarbons can be converted to hydrogen through steam reformation. Methane plus water produce carbon monoxide and 3 molecules of hydrogen. Carbon monoxide can be converted to hydrogen through water gas shift reaction. Carbon monoxide plus water produce carbon dioxide and hydrogen. Besides gaseous products, bio-oil from pyrolysis can also be used for hydrogen production. For this, bio-oil is first divided into two fractions based on their solubility in water. The water soluble fraction is used for hydrogen production through catalytic steaming reforming. The next is gasification. Gasification involves the partial oxidation of biomass components at high temperature above 1000 Kelvin and in the presence of oxygen to yield hydrogen, carbon monoxide, carbon dioxide, methane and other hydrocarbons and charcoal. Charcoal is later converted to gaseous products. Biomass plus heat in the presence of steam yield hydrogen, carbon monoxide, carbon dioxide, methane, hydrocarbon plus charcoal. Similar to pyrolysis, the gaseous products are converted to hydrogen through steam reformation and water shift reactions. Biological methods. The following are the major biological methods involved in biohydrogen generation. Biophotolysis, biological water gas shift reaction, photofermentation, dark fermentation and microbial electrolysis cells. Let me see in detail about biophotolysis. Biophotolysis involves the splitting of water to hydrogen using the photosynthetic mechanism of plants. Generally, microalgae are utilized for this purpose. The method is classified into two types: direct biophotolysis and indirect biophotolysis. Direct biophotolysis. Direct photolysis is observed in green algae and cyanobacteria. This happens due to the presence of hydrogenase enzymes which is absent in higher plants. When the photosystem 1, that is PS1, absorb light, 2 electrons are released along with the split of water to generate proton ions and oxygen. Ferredoxin, electron carrier, accepts the electrons from photosystem 2 using the solar energy absorbed by photosystem 1. These electrons are transferred to hydrogenase which convert hydrogen ion to hydrogen. Indirect biophotolysis. Biohydrogen generation through indirect biophotolysis is usually carried out using cyanobacteria. The process involves the following steps. Step 1. Glucose is produced through photosynthesis using carbon dioxide. Steps 2. Glucose is converted to hydrogen and acetate through aerobic dark fermentation. Acetate is later converted to hydrogen. Biological water gas shift reaction. Convention water gas shift reaction involves the reaction of carbon monoxide with water to yield carbon dioxide and hydrogen. Some photoheterotrophic bacteria such as Rhodospirillum species able to carry out the same process under anaerobic conditions. They use carbon monoxide as carbon source to generate ATP which results in the production of hydrogen. Carbon monoxide plus water produce carbon dioxide and hydrogen. Gram-negative bacteria, Rubrivax gelatinus, and gram-positive bacteria, Carboxydothermus hydrogenoformas, are other examples for microorganisms which can carry out the process. Photofermentation. Photofermentation involves the conversion of organic substances present in the biomass to biohydrogen by a diverse group of photosynthetic bacteria including Rhodobacter spheroides, Rhodobacter capsulatus, Rhodobacter sulfidophilus, Rhodopseudomonas palalistis, and Rhodospirillum rubrum. The process is known as photofermentation since it is carried out in the presence of light. The process is similar to biophotolysis, however, it does not involve the splitting of water. Instead, electrons are obtained from organic acids such as acetate, lactate, butyrate, maltate, etc. Ferredoxin transports these electrons to the nitrogenase from where protons are converted to hydrogen. The major advantage of photofermentation is that industrial or agricultural waste can be used as substrate for hydrogen production and thus benefits for efficient waste management. Dark fermentation method of hydrogen production. Dark fermentation is carried out by fermentative microbes without using solar energy. These microbes can be facultative or obligate anaerobes. These microbes utilize organic substrates such as glucose to produce hydrogen and carbon dioxide as the major products. Along with hydrogen and carbon dioxide, some amount of other gases such as methane, hydrogen sulfide, etc. are also produced depending on the reaction process and the substrate used. If glucose is used for fermentation, acetate and butyrate are produced along with hydrogen and carbon dioxide. The process can be represented through the following equations. Glucose plus water produce acetic acid, 4 mole of hydrogen and 2 mole of carbon dioxide. When the acetate is obtained as a product, 4 mole of hydrogen are produced from 1 mole of glucose. Glucose combined with water, it produces butyric acid plus 4 mole of hydrogen and 2 mole of carbon dioxide. When butyrate is obtained as a product, 2 mole of hydrogen are produced from 1 mole of glucose. However, it is practically impossible to obtain 4 mole of hydrogen from 1 mole of glucose since both acetate and butyrate are obtained as end product. Hydrogen production through dark fermentation depends on the following factors. pH. pH range between 5 and 6 is normally maintained for optimal hydrogen production. Hydraulic retention time. This measures the average time for which a compound remains inside the reactor. It has been reported that hydraulic retention time of half day can provide maximum hydrogen yield. Hydrogen concentration. When the concentration of hydrogen increases beyond a specific value, the process shifts to produce more reduced substrates such as lactate, ethanol, acetone, butanol or alanine. The next is microbial electrolysis cells. A microbial electrolysis cells consist of an anode, cathode and a membrane in between them. Electrochemically active bacteria are present in the anode. These bacteria oxidize the organic substances to produce carbon dioxide, electrons and protons. Electrons are transferred to the cathode through external circuit, whereas the protons move to cathode through the membrane. A small amount of external electric current is added to the cathode along with the electrons from the microbes to improve the efficiency of hydrogen production. At cathode, protons are converted to hydrogen using the electrons. The membrane is used to improve the purity of hydrogen produced as well as to prevent the consumption of hydrogen by bacteria.